Hello, everyone. After Olga's talk, I will now dive right into your project of whole genome sequencing. And I will start with the very first steps, which are sampling your organism of interest and what to do with it as you are sequencing and before you sequence it. And my name is Paco Cárdenas, and I work at the Museum of Evolution. I'm responsible for the collections of zoology here at Uppsala University. So regardless of the environment where you will be collecting your organism of interest, you will need to have basic good practices and think ahead before even collecting the organism that you want to sequence. And the very, very first step that you should do before collecting even is getting all your documentation in accordance to the rules and regulations of the country where you're going to collect. And this could be from buying a fishing permit to getting an official government permit and so on. And for all of these questions, I redirect you to one of the following talks. And of course, now you have the documentation, you're going to start to collect, but what to collect? It's not only the organism that is of interest, it's also all the data around this organism, what we call the metadata. And this metadata can be very rich and diverse, but there are essentially three pieces of information that we absolutely need to have. And that is the name of the species, where it was collected, and when it was collected. These are the three pieces of information, the three basic pieces of data that we need before when you're collecting your specimen that you should record. Of course, most of the scientists, they record much more. And I will give you a few examples. But let me just stress one point here that I want to make, and that is the identification of your organism. You're going to produce so much data with your whole genome that, of course, you don't want to get that very first piece of information wrong. You want to have the right name on the specimen that you're collecting. This is very important. Don't be afraid to ask the help of a taxonomist to get the name right. Once you have the name, the locality, and the date, there are other pieces of information that are interesting, and that could be super relevant once you have your data. For instance, the sex of your organism, what gear you used, who collected it, who identified it is also very relevant. The habitat, what characters your organism has, how did you fix it, what were the environmental parameters around your organism, and so on. I always recommend to photograph your specimen even before collecting it. If you can have a picture in situ, it can be very useful with a scale, of course. If not, any picture of the specimen will do and will need to be published along with the whole genome information and sequence. Label your samples always from the very start, from the moment you collect it. You can use a field number, you can use pre-printed labels. A lot of people now are using QR codes, pre-printed QR codes, as you can see here on the pictures. Please use good quality paper, what we call archival paper that is a bit thicker, and pencil. Pencil will survive if you put it in ethanol or in other types of fixation. So pencil is always the best. Avoid pens or ink. Once you're on the field, for the same organism, sometimes you will need to subsample for different types of studies. Some people will want to do whole genome sequencings, but maybe some people will like to get additional data from that same organism. That could be metabolomics. It could be transcriptomics, just like Olga also said, having the RNA-seq is important for the whole genome sequencing. But for every one of those, you will want to use maybe different types of fixation, from liquid nitrogen to RNA letter to freeze drying. Of course, when you subsample, please keep track of all of your samples and label the subsamples accordingly. It's very important to keep track. And while you do that, always remember that in the end, you will need to keep a voucher of your organism. And I'm going to emphasize and stay on that idea for a while. But before that, I also want to stress the different types of equipment once your organism has been subsampled that can be used to ship that specimen to the sequencing facility. These are essentially dry shippers where you can put liquid nitrogen and your organism of interest. And these can go on an aircraft. You have different sizes. That's the very best option. Otherwise, you can always use dry ice. It will keep your organism at minus 80 degrees for about 24 to 48 hours. Voucher deposition. The importance of vouchers has been raised many, many times, way before people did genomes. Just because when you get data from an organism, there could be several steps in which mistakes are done. And keeping a voucher enables you to come back to the original specimen and revise either the identification or the characters that were noted. So it enables you to come back and reproduce some of the data that is tied to your dataset, and that is very important. There's been many papers stressing this, the critical importance of vouchers in different fields, and notably in genomics. I will show you one example of an article where they looked at the percentage of genomes that were tied to an actual voucher specimen deposited in a museum or an institution. And they took the case of vertebrates, and they show that only 11% of vertebrate genomes actually had a voucher specimen. And you might think, vertebrates, we know all of them. It's not that complicated. It actually is. The taxonomy of many vertebrates is still confusing, and it's still being revised. I'll give you 3 examples, but before that, I just want to stress why it's important to keep this voucher. It will make your data more reliable. It will make your data more reproducible, and it will make your data accessible to other people that want to double check it, for instance. And some people might argue, well, we have all this genomic data, and that's probably enough to identify the species. But you're probably relying your identification on gene bank sequences, previous gene bank sequences, and there could be many errors there as well. So keeping a voucher is probably the best step you can take. One example why it could be important is the example of the electrical eel genome that was published in 2014. A couple of years later, it was shown that this species is actually a species complex. Unfortunately, no vouchers were kept for the genome, so it's very difficult to know what was actually sequenced in 2014. Another example, a massive fish, the sunfish, Mola mola, the genome was published in 2016, and 2 years later only, it was shown that it's actually a species complex with 3 different species. Unfortunately, no vouchers were kept, so we don't know which one of these 3 different species was actually sequenced. A third example is the one of sponges, and I think it pretty much shows what often happens either in tropical environments or in marine environments where a lot of our organisms grow on each other. And if you don't pay attention, you could actually sequence not just one organism, but 2. And you cannot actually be sure what you sequenced. And here I'm showing pictures of a sponge. It looks yellow. When you cut it in 2, it's kind of yellowish, brownish inside. These are actually 2 sponges. one yellow sponge growing on top of a brownish sponge. So when it was sequenced, everything was put together, and two totally different species were actually sequenced. By keeping a voucher, we can come back and actually double check what has been done, what has been collected, and re-identify those species. Very important to keep vouchers. Of course, there will be cases where the organism of study is far too large, far too small. There will be challenges in having a voucher for everything. But there are solutions where you can deposit a proxy of a voucher. It can be just a piece of a specimen that is representative of the specimen. It can be a specimen from the same population, for instance. But the best thing is also always publish the picture of the organism that you actually sequenced, as well as the description of the organism you sequenced. Where should the voucher be deposited? It's no use to keep a voucher and keep it in your lab and say, okay, I've done my job, because that voucher is not accessible to everyone. It's not searchable. It's not necessarily well preserved. So it's actually not reusable for anyone. So when you think of depositing a voucher, having the voucher is not enough. You need to think of depositing in a place where it will be findable by the scientific community, accessible by everyone, reusable if people want to get more data out of it, and it will be preserved forever, basically. Where can you do this? Also in a place where you can preserve all the permits and the documentation. And this is the kind of place that we call a museum. Museums do that. They preserve archives as well as specimens, organisms. And there's 4 major museums in Sweden, for instance, and that's the Museum of Natural History in Stockholm, the Museum of Evolution in Uppsala, the Biological Museum in Lund, as well as the Göteborg Natural History Museum. All of these places will very happily accept your voucher, will give you an accession museum number, which will make your voucher searchable, accessible, because this voucher then can be sent on loan to other scientists, and it will be preserved forever, along with all the documentation linked to it. Since I work at the Museum of Evolution, I just wanted to show you this Museum of Zoology that we have here, that was founded essentially by Linnaeus in 1743. We currently have more than 5 million specimens, more than 6,000 specimens that are connected to Linnaeus, so that we call them Linnean specimens, and more than 41,000 types, so original specimens from new species. Our collections are on the Evolutionary Biology Center campus, in different parts of the buildings, depending if we're speaking of the collections of zoology or the collections of botany. We also have collections of paleontology and mineralogy. And these collections are very, very diverse, and they range from wet preserved specimens to dry preserved specimens. They can be whole organisms, or just the dry shells, or the eggs, and so on. These collections are curated, they're managed by what we call curators, and depending on the specimen you want to deposit in our museum, you will contact one of these experts that will take care of your specimen. So we have curators for lichens, for fungi and algae, for higher plants. I'm the curator for zoology, for instance, and we have also 2 other curators for entomology, insects. So you're giving your voucher to the Museum of Evolution, for instance. In exchange, we give you a museum accession number. You will use that museum accession number to put it in the publication of your whole genome or of your article. So here's an example where we sequenced the whole genome of a deep-sea sponge, Geodia bereti, and in the material and methods, we have inserted a sentence saying the voucher is deposited at the Museum of Evolution under the following number, so that anyone can easily find it, come to the museum, or ask for a loan of that particular specimen that they want to revisit or study further. Just to show you that how important vouchers are, I also want to show you this article that we published a few years ago on the good practices in natural product studies. In chemistry, it's also very important to have vouchers because you want to know where the chemical compounds were extracted from. And in this paper, we have looked at vouchers that were kept of sponges where people have claimed they have isolated some particular triterpenes, which are lipids. I've been looking at 44 articles spanning 1981 to 2022. 7 articles didn't have any voucher, so there's no way you can actually check these identifications or results. In total, in these 44 articles, I could gather 27 vouchers, compile a list of 27 vouchers. All of the ones that were deposited in museums were accessed, and I could revise them. But several specimens that were deposited in other places than museums were actually lost, and I could not access them. In total, I could access 21 vouchers, but over these 21 vouchers, more than half were misidentified. So they actually needed to be revised. And I would say overall, more than 65% of these studies actually published with an incorrect species name. So that's the extent of errors you can have in the literature. And to check and correct these errors, you need a voucher. And that is the take-home message. I hope you will remember the importance of vouchers and these basic good methods that you need when you prepare to collect a specimen for whole genome sequencing. Thank you very much.